Thank you, Abby. Hello, everyone. Welcome to the webinar. I'm glad you've joined us. Today, we're going to be discussing photosynthetic survey and response curve measurements on an introductory level. So let's start with some basics. Photosynthesis is a plant's use of light and CO2 to create sugars, which can then be used for growth and development. But we can't really measure photosynthesis directly. Instead, what we do is measure photochemistry, and we use that as a proxy for photosynthesis. We can do this in two main ways. We can look at gas exchange, or we can look at chlorophyll fluorescence. This webinar will only discuss techniques that use gas exchange. But if you're interested in chlorophyll fluorescence, we have several webinars that discuss it, and I encourage you to watch some or all of them. Among the many things you can do to measure photochemistry are two broad categories that we're going to call survey measurements and response curves. So, what are they? What do they tell us? And how do we do them? The how is a bit more complex because it really depends on your plant and your research question, but we'll discuss the hows very broadly. So first, let's talk about survey measurements. Survey measurements tell us what a plant is doing at any given point in time. You could follow a plant or an individual leaf through a growing season, through a single day, or both. This graph, for example, shows the data from a research paper in which they looked at the response of soybean leaves over the course of a day, which is called a diurnal measurement. The plants in this case were exposed to ambient or elevated carbon dioxide or ozone, and you can see that their response changed over the course of the day, depending on the treatment type. By looking at the plant's response to the various treatments over the course of the day, we could develop some hypotheses as to what might be driving that response. It could be nutrients, or water availability, or temperature, or maybe something else entirely. Response curves, on the other hand, tell us how a plant responds to a specific environmental variable. All the other variables are held constant as best we can. Response curves allow us to measure how a plant responds biochemically and physiologically within the living leaf. We can then correlate those measurements with information about growth, development, or even other survey measurements. Now let's go ahead and do a little poll. I'm curious, to find out whether you're interested in survey measurements, response curves, both, or maybe you don't know, and that's why you're watching this webinar. Abby's going to put a poll up on the screen, and we'll give you a few minutes to answer.
Okay, Elizabeth, here's our poll question. Are you interested in survey measurements, response measurements, both, or I don't know? We'll give everyone a few minutes to answer. Thank you, everyone, for answering. Those responses are very interesting. Elizabeth, I'll hand it back over to you.
Okay, as usual, we have an interesting mix of responses. Almost always happens with polls. So let's move on and talk about some key elements of survey measurements. The main thing to remember is that survey measurements are fast. They're also called snapshot measurements or quick measurements or spot measurements, and they should be completed at under 5 minutes per plant. To do this, we have to work at the plant's ambient environmental conditions. If we don't, the plant will have to acclimate to the new environment, and that means we have to wait for it to do that, and it's not a survey measurement anymore. When it comes to environmental control, we have 4 main options in the 6400. CO2, temperature, H2O or flow, and light. So what do we want to do for a survey measurement? Well, reference CO2 is the fastest, so that's what we want for a spot or survey measurement. Be sure to check the ambient CO2 concentration so you know where to set the reference CO2. In terms of temperature, we want temperature to be ambient, but keep in mind that we do have a diurnal temperature cycle, and it will probably have an impact on your plants. You might consider setting the temperature to a constant value, which would warm your plants up a little in the morning and cool them down in the afternoon. Or you could just use the ambient temperatures and be aware of the Arrhenius equation and its relationship in plant behaviors. In terms of H2O control, we're going to use a constant high flow rate to flush the chamber quickly so that when we open and close the chamber, we can get back to an ambient, steady state inside the chamber very quickly. Watch your relative humidity, though, and keep it between 50 and 80%, and we'll discuss more about that on the next slide. In terms of light, we want to use ambient light, and we have two options here. We could use the LED light source and the external quantum sensor to make sure that we are at ambient, or we could use the clear top chamber. The clear top chamber is great for survey measurements because you make sure you're working at ambient light and you also save a little battery life. But you need to make sure that you keep the leaf at the same plane as your light source, whether that's the sun or a growth chamber's light or something like that, because otherwise you might shade your leaf, and we want to avoid that. Now, I mentioned ambient water. Spot measurements or larger diurnal measurements depend on really working at ambient conditions, so we don't have to wait for it to acclimate to something. The one exception to our ambient rule is water. Normally, plants have a boundary layer around them, so that even if the outside air is 30% relative humidity, the boundary layer near the stomates and the leaf is closer to 100% relative humidity. The LI-6400 mixing fan removes part of this boundary layer, so we have to add the humidity back again. If we don't, the stomates will close and we won't see any gas exchange. However, we still want a gradient between the inside of the leaf and the outside of the leaf, or we won't detect a change in response. So, avoid the first picture, which is less than 50%, and avoid the last picture, which is more than 80%. This will make your survey measurements much faster and much more stable. Speaking of stability, how do you decide when you should log a survey measurement? Well, if you look at this graph, you see that photosynthesis and conductance are at a steady state across these two graphs, which means our overall slope is 0. There are little bobbles up and down, but look at our scale, especially for conductance. It's tiny. The little changes aren't the important part. The slope is the important part. It would be safe to log in this case. You can also look at stability criteria and total CV on line E of new measurements mode, but remember that the instrument's stability criteria are only as good as you make them. Let's change gears a little bit and start talking about light response curves. Light response curves can provide measurements about how plants qualitatively and quantitatively utilize light energy. Light response, which is also called AQ curves, are used to determine the light compensation point, also called LCP, and light saturation point, which is abbreviated LSP. Plants that are grown in sun or shade will have different LCPs or LSPs, among other changes in their physiology. Using a light response curve, we can also determine ASAT, which is photosynthesis at saturating light, and RD, which is respiration. Now, ASAT and RD values are very specific to each plant and each growing condition, and the numbers you see on this slide are just an example of the values that we saw with this particular plant. However, the overall shape of the curve is fairly standard. So what are we looking at when we look at a light response curve? Well, we're focused on the light-dependent reactions here. When a plant reaches the light compensation point, there's enough light to successfully complete the light-dependent reactions you see on this slide. Since the light-dependent reactions produce ATP and NADPH, which we need for the carbon reactions, net photosynthesis can't begin without the light reactions going through successfully. When the light saturation point is reached, light is no longer the limiting factor for photosynthesis. Maybe it's nutrient availability, maybe it's water, maybe it's CO2. Whatever it is, it's not light. So light response curves can be done in two basic ways, although, as usual, there are endless variations within these two ways. A fast light response curve starts with a light-adapted plant and goes down to 0 light. Light is off. This typically takes 30 minutes or so, depending on how many set points you have and how long you spend at each. A slow light response curve starts at 0 and goes up slowly to a saturating light level. Plants respond to reductions in light much faster than they respond to increases in light. Fast light response curves allow you to do many curves in a single day, but the stomatal conductance values at the lower end of the curve are often too large because the stomatal processes haven't fully acclimated to the light level they're experiencing. Many things in the plant happen on different scales, and so we might find that one thing was stable, but another thing was not quite where it would be if it was at true equilibrium. Slow light response curves allow you to make sure that the plant is fully acclimated to each level in every way, but since each level can take up to 30 minutes, only a few curves can be done in each day. If you're interested in stomatal conductance at or around the compensation point, a slow light response curve is probably best. You might also be interested in quantum yields, and in that case, you wouldn't do the whole curve, you'd only look at a portion of it. Remember that your research question will direct the way you do a light response curve as well as the points you set within it. Let's talk for a moment about autoprograms. Autoprograms on the LI-6400 XT allow the computer in the instrument to do what it is good at, while we do things that we are good at. An autoprogram has a series of commands that we ask the instrument to do over a period of time. Think of it a little like cruise control on your car. Your car is very good at determining how much gas is required to travel at 50 miles per hour, but you are still in the driver's seat. Your hands are still on the wheel. It's similar to the autoprogram. We let the computer do what the computer is good at, and we focus on whether the numbers we see on the screen or in the graph are reasonable and logical for our experiment. The newest version of the software, which is available for free on our website, has a few nice features for the autoprograms. The main one is that all the details are on the same page. In previous versions, you would answer questions successively, which meant if you typed an incorrect value, you'd have to stop the autoprogram and start over. I've done this many times. In the new software, you can be sure all your settings are correct, and when you're happy with them, you can go ahead and press Start. In terms of environmental control for a light response curve, it's going to be very different from what we do for a survey measurement. We're going to control everything a lot more carefully because we want to make sure that light is our only real variable. So for CO2 control, we're going to control sample CO2, and we need to make sure that we set our CO2 to ambient. Otherwise, we're doing a nested CO2 and light response curve, which you can do, but that's not a simple light response curve at that point. You're answering a different question. If your temperature is at all changeable, control leaf temperature to a constant value. If you're working in a temperature-controlled environment like a growth chamber, you can skip this. Otherwise, you do need to keep that in mind. For water control, we're going to use a constant water mole fraction and let the instrument adjust the flow rate for us. This will make sure that the plant's stomatal response isn't influenced by changing water levels. And of course, light, well, that's what we're changing. We're doing a light response curve. You can either use simple PAR settings in a fluorometer, an RGB light source, or an O2B, or you can move within a canopy if you want to use a clear top chamber. Clear top chamber canopy light response curves are a little messier, but they are certainly possible. So let's move on to CO2 response curves. Just like light response curves are the plant's response to changing light levels, CO2 response curves measure the plant's behavior when CO2 levels are changed. When we do a CO2 response curve, which you'll also see referred to as an ACI curve, we're looking at the other half of the photosynthetic mechanisms in the leaf. Rather than looking at the light reactions, now we're looking at the carbon reactions. And this image is the carbon reactions called the Calvin cycle, or Calvin-Benson-Basum cycle, or reductive pentose phosphate cycle, and of course the other PCR. Three main biochemical parameters are obtained from a CO2 response curve, although there are several more, but most of the time people look at these three. They look at Vc-max, which is the maximum velocity of carboxylation of rubisco. That's the first step in our pathway on the right. And they look at Jmax, which is electron transport for RUBP regeneration, which is our last step. The other thing they look at is triose phosphate utilization, which is the transport of the sugars created into metabolic pathways. We can also look at the CO2 compensation point, which is going to be very different for C3 and C4 species due to CO2 concentration mechanisms in C4 plants. And so an ACI curve for a C3 plant and a C4 plant will look completely different. This, for example, is an ACI curve for a C3 plant. And there are many different ways to do an ACI curve, as well as doing a light response curve. Some ACI curves start at saturating CO2 and go down. Others start at the bottom, 0, and go up. Others start in the middle and go down. Which one you do depends on the research question you're trying to answer, as usual. Things to keep in mind, though, are that at higher CO2 concentration, the stomates close because there's plenty of CO2 in the environment and the plant can reduce possible water loss but still take in plenty of CO2. So it just kind of relaxes a little bit. At lower CO2 concentration, there's an effect on rubisco. Rubisco might be deactivated in some way, or oxygen might outcompete CO2 for the rubisco binding sites, especially in a C3 plant. Rubisco is quite happy to bind with oxygen as much as CO2. Sometimes it's happier, in fact, and so if we don't have a lot of CO2, we will have oxygen bound to it instead. If you're interested in the CO2 compensation point, you'll probably focus on the lower end of the curve. If you want to know about Jmax or TPU, you'd focus on the upper end of the curve. How you set your concentrations will be affected by these choices. Either way, determine what question you want to answer and start from there, rather than assuming that just because one paper or one researcher did it this way, or even that you see it in our manual done this way, you should do it that way. It will all depend on the question you're answering. We have an auto program for ACI curves too, and it's just like the auto program for the light response curves. You'll set all of your parameters and then press start. One thing that's important to note when it comes to an ACI curve is that you have an option to control matching frequency during an auto program. And you should set the match option to always for a CO2 response curve. It's very important to match when we change CO2 concentrations so significantly, and I'll discuss that in a few slides. And again, This is just an example of how to set up the measurements, so don't assume that the numbers you might see on these slides are the numbers you should use. Our environmental control options for a CO2 response curve are again focused on not changing anything but CO2. So, for CO2 control, we're going to use reference CO2. That's fast, and it's going to make it easier for us to adjust to our different set points. For temperature, again, if it's at all changeable, control leaf temperature to a constant value. You might be able to skip this if you're working in a temperature-controlled environment. We're going to use a constant water mole fraction, so the instrument adjusts the flow rate for us for this response curve as well, ensuring that our plant's stomatal response isn't influenced by changing water levels. And for light, we're going to use a constant saturating PAR level, which is going to be easiest and fastest if you use a supplemental light source. But it is possible to do it with just the sun. So, what are the benefits of each measurement, and how do you decide which one to do? Well, a survey measurement gives you a quick snapshot of a plant's behavior. It's fast, it can be used to explore seasonal or diurnal plant responses, and it can be used to explore treatment responses. A response curve looks at varying environmental conditions. It can be used to understand leaf biochemistry and plant physiology, and you could repeat it through the course of a season to record biochemical trends. Most often, people combine response curves and survey measurements because we do learn different things from them, and you might want to know both things. Which one you end up doing and how often you end up doing it will depend entirely on what kind of question you're trying to answer. One thing I want to mention is that whether you do a response curve or a survey measurement, you want to make sure that you go through the checklist in Chapter 4 before any measurement. The checklist in Chapter 4 provides us a general hardware and software overview. If you go through it each day, you can have confidence that your measurements are not being influenced by any problems with hardware. If you don't go through the checklist, you might discover you had a problem when it's too late to fix it. And no one likes discovering that they can't use data. The other thing I'll mention is the importance of matching. Matching flows the same air through the sample and the reference analyzers, which helps us detect and then correct any analyzer drift in the sample cells. This could be caused by changing CO2 concentration, changing temperature, changing flow, dirt, pollen, really anything that makes the analyzers sort of wander off from each other. We designed our instrument to take the analyzer drift into account, so it's very important that you match and make sure that there isn't any. So when do we match? We want to match when we start. We want to match when delta CO2 or delta H2O are small. Small is usually defined as less than 10 ppm for delta CO2. We want to match when flow rate changes more than 100 micromoles per second. We want to match when CO2 concentration changes more than 100 ppm. We want to match when the temperature changes more than 5 degrees C. And if none of those is true, we want to match at least every 20 to 30 minutes. It's difficult to match too often if you are matching correctly and not getting any error messages. My protocol in graduate school was that we matched every single plant, so that would be 2 to 3 minutes between matches. As long as you're not getting error messages, that's fine. So I hope that this webinar has provided you with a general idea of what survey measurements and response curves are, and what you could do with them and how you could use them. If you have more questions about your specific application and what measurements you should do, you can let us know. We can't always tell you exactly how to set up your experiment. That's somewhat your job, but we will be happy to give you ideas of what we have learned in terms of best practices with the instrument. So if you do have any questions, please feel free to contact us. Speaking of questions, Abby, do we have any questions from the audience yet?
Yes, Elizabeth, we did get a few questions. The first question is, how do I analyze data from a response curve?
Oh, that's a tough one because everybody likes to do it a different way. There are Excel spreadsheets that will help you analyze the data. There are packages in various software programs. We know that people use MATLAB or SPSS or R. Really, it's something that you will figure out. And we don't have any specific recommendations, but more than likely, as you read through the literature, you'll see how other people have done it and make the decision from there.
Great. The next question is, do I need to log multiple times for survey measurements?
Yeah, we see this a lot in data files. People will be clamped onto the same leaf and they will log 4 or 5 times, maybe 10 seconds apart. And actually, I was trained to do the same thing, but you don't really need to do that. The default settings in the 6400 XT is for a 4-second running average of any data point that you see. So anything you see on the screen or see in the log file, that's a 4-second running average rather than just a moment in time. And within that 4-second running average, if you have the newest software version and the newest hardware version, we have about 300 points per second. So every logged value is up to 1,200 points, which gives you a lot of power. Most of the time, you can just log once on one leaf and then go to the next.
Okay. Another question is, can I do temperature response curves or water?
You can. You can do a temperature response curve, a humidity response curve, a VPD response curve. You can do a response curve for all of the environmental variables. But I will warn you that temperature response curves and water response curves are a little bit more complicated, and they do take a longer period of time, and you'll often have to have a growth chamber to give you a really wide range of temperatures or water settings without causing problems with the instrument. So more equipment is required, but you can do it.
Okay. Well, that's all the time we have for live questions. If there are any outstanding questions, our panelists will be sure to answer those before we sign off. Thanks, everybody, for attending, and thank you, Elizabeth.